Hi, everyone. Thank you so much for coming to my talk today. My name is Haley Plaas. I'm a PhD student at UNC Chapel Hill. And today I'm going to be talking to you about my project that examines cyanobacterial toxins, especially microcystin, in aerosol produced in the airshed of the Chowan River-Albemarle Sound in eastern North Carolina. So without further ado, cyanobacterial blooms occur in freshwater and estuarine ecosystems, and blooms are classified as harmful when they produce toxins known as cyanotoxins, which are linked to a suite of adverse health outcomes. One toxin that's commonly produced in many systems across the world, including the Chowan River, is microcystin, which is a potent liver toxin, and humans might be exposed to microcystin via drinking water, shellfish tissues, or through skin contact, or when swimming, or even via inhalation of lake spray aerosol. And less is known about the health effects from the inhalation of cyanotoxins in spray aerosol, and this is partially because of a lack of data demonstrating environmental concentrations of cyanotoxins like microcystin and aerosol. So there are many anecdotal cases reported about people experiencing respiratory and eye irritation during bloom conditions, so there might be chronic health impacts associated with the inhalation of cyanotoxins that we are not yet aware of. And as CyanoHABs continue to proliferate across the globe, more research is published on the production of spray aerosol in freshwater and estuarine ecosystems. It's increasingly important that we gain a strong understanding of the incorporation of these toxins into aerosol, especially during bloom conditions. So in eastern North Carolina, our study site, the Chowan River-Albemarle Sound, is an estuary that has seen many recent summers of CyanoHAB activity, chiefly composed of Microcystis, which is a genera of cyanobacteria that frequently produces high concentrations of microcystin. So you can see in this map here, published by the North Carolina Department of Environmental Quality in 2019, all of the reported CyanoHABs across the state are here in red. And there's a heavy concentration of those toxins those cyanobacterial blooms being recorded in the Chowan River. And furthermore, the Albemarle Sound stays very fresh due to the Outer Banks Formation not allowing a lot of exchange with the Atlantic. So CyanoHABs can thrive in this oligohaline system. And this is an important area for tourism, recreation, and fishing in the state. Last summer, the microcystin concentration was reported up to 620 micrograms per liter of microcystin in some locations, which is 60 times more than the World Health Organization's recommendations for swimming. Plus, the Albemarle Fetch is up to 14 miles wide in some areas, allowing for significant wave fields to develop. And so there is a lot of breaking wave action for primary aerosol production here at the water-air interface. So for my purposes, I just refer to the production of lake spray aerosol or sea spray aerosol as spray aerosol, since there is no specific term for estuarine aerosols. And so this figure here depicts some of the interacting physical, chemical, and ecological processes which may interact to promote the incorporation of cyanobacterial cells and their chemical constituents into spray aerosol. So cyanobacterial cells and their toxins are positioned perfectly to be potentially interacting with air bubbles that are entrained via wind and wave action. The first aim of our study was to survey the concentration of microcystin in aerosol and water and examine cyanobacterial DNA in spray aerosol as well. So we are looking at water samples in parallel for comparison, and we'll calculate enrichment factors in aerosol and examine the cyanobacterial species and congeners of microcystin that are preferentially aerosolized. The second aim of our study is to examine the environmental correlates of cyanobacteria in aerosol. So we're evaluating dynamic weather conditions, which may promote the aerosolization of toxins, as well as some of the water quality parameters that might impact cyanobacterial ecology. We've recorded ambient meteorological conditions that influence spray aerosol formation and transport, such as air and water temperature, relative humidity, wind speed, wind direction, sunlight, etc., as well as some ambient water quality parameters such as turbidity, temperature, pH, and salinity. To carry out these objectives, we had aerosol samplers deployed at 2 field stations that we set up with the help of a group of community scientists in the area. And our campaign ran from June to the end of September, which is during peak cyanobacterial bloom season. To collect aerosols, we used high-volume impactors with cyclone inlets to sample PM2.5 onto 103 millimeter quartz fiber filters. We collected both single-day and 2-week integrated aerosol samples. These 2-week samples consisted of collection for 12-hour periods from 7 a.m. to 7 p.m. over the course of 2 weeks. And the single-day samples were collected in 7 to 8-hour periods, and these samples are being used to calculate those enrichment factors from the water concentrations that we took at the same time. So to assess spray aerosol production and to source the PM2.5 appropriately, we measured real-time mass concentrations of PM2.5 with a nephelometer, and we are using these concentrations with wind speed measurements to estimate the relative spray aerosol production. Water samples were collected on filters or in aliquots, and those water samples will be analyzed for cyanobacterial DNA, intra and extracellular microcystin, diagnostic algal pigments, chlorophyll A, cell counts, and nutrients. And aerosol filters will be analyzed for cyanobacterial DNA and bulk microcystin concentrations. All of our filter extracts and samples in the lab will be analyzed with a targeted approach. Microcystin will be quantified via HPLC MS, and we're using the triple quadruple mass spectrometry, and we're quantifying 10 microcystin congeners. Cyanobacterial DNA will be analyzed via amplicon sequencing with primers for general bacteria 16S rRNA, general cyanobacterial 16S rRNA, and then microcystin A, which is an encoding gene in cyanobacteria for microcystin. Cell counts will be conducted with flow cytometry, and our diagnostic algal pigments are being evaluated with high-performance liquid chromatography, and we'll also be looking at biomass proxies via chlorophyll, and we'll be looking at total, dissolved, and particulate nitrogen and phosphorus with flow injection analyses. So of the water and aerosol samples we've extracted thus far, and in agreement with the routine monitoring data from the state, there has been very little microcystin detected in the system this summer, which is quite divergent from past years. And we attribute this to the type of cyanobacteria which bloom this season. So past summers have been marked by that Microcystis growth, which is that genus of cyanobacteria that frequently produces microcystin. But this year, the blooms have been dominated by Dolichospermum in the early summer and Cylindrospermopsis in the later summer, and neither of these frequently produce microcystin. So what we're doing is we're analyzing all of our water samples originally for toxins and DNA before we extract any of our aerosol filters, because we don't want to waste our precious aerosol filters on toxin analyses if the toxin is not in the water to begin with. So we hope to have these toxins quantified within the next month so that we can begin analyzing our aerosol filters. This line graph here also just gives a little demonstration of some of the raw data from PM2.5 mass concentrations we've measured in the air at each site over the course of June. And we've marked some anomalies with probable explanations for spikes in the ambient mass concentrations, which were not correlated for wind speed and direction. The microcystin concentrations are likely low this summer, so our backup plan is to quantify other toxins, such as anatoxin, which is more readily produced by Dolichospermum. And we're using some extra water filters that we have collected in this case to examine for these other toxins. For the August samples, we're going to analyze for Cylindrospermopsin, which is a toxin that's commonly produced by Cylindrospermopsis, which was present. And whichever toxin is more prevalent in the water samples, we'll attempt to quantify and detect in the aerosol samples that are correlated to those water samples. So even if the toxins are not produced at all this summer, we'll still be analyzing for cyano DNA and PM2.5 this summer, which may provide evidence that CyanoHAB cells or cell fragments are capable of incorporation into spray aerosol. More research should aim to understand the form in which cyanotoxins exist in aerosol. That is to say, are they transported within cells or in dissolved form? And we speculate that processes which may promote cell lysis and increase dissolved toxin concentrations may lead to higher concentrations of toxin in aerosol. So CyanoHABs nearing senescence or treated with algicide or occurring along estuarine gradients such as the Chowan River might contribute the greatest to cyanotoxin enrichment in aerosol. And with that, I just wanted to say say thank you so much for listening to my presentation. I hope in a future meeting I can present some more complete findings. But thank you to everyone who has supported me to make this project possible. And for any listeners with questions, I look forward to chatting with you in the chat box for this virtual event.